So good afternoon, everyone. We are going to start a new CCMAR seminar. Today we have Mark Tarasco, which is a PhD student at CCMAR in the Bioscale lab. Mark, during his master thesis, developed a protocol using Operculum to study osteotoxicity, to evaluate osteotoxicity compounds. And now Marco, based on that research, is doing his PhD here at CCMAR. And he's gonna talk to us about his current work. Besides that, Marco also developed an image toolset that he recently published in Bone Journal, and he will include it in this presentation. So Marco, the stage is yours. Whenever you want.
Thank you, Jill. I will share my screen. Can you see it?
Yes.
Yes.
Okay. Hi, everyone, and thanks for joining today's seminar. I have divided this seminar in two parts. In the first one, I will show you some of the Zebrafish in vivo system, which we use currently in our lab to assess osteotoxicity, and we will see the example of benzolfapyrine. And in the second part of the seminar, I will talk about image analysis tools for automated data processing. I will start first with a little introduction. We know that the vertebrate skeleton is very important among all vertebrates, including us. First, because it gives support to our body. The skeleton is a mineral reservoir of calcium and phosphate. In mammals, it is source of blood cells. And of course, protect our internal organs and also allow us to move. It is mainly composed by bone and cartilage, and in our lab, we focus mostly on bone. Bone is composed by a mineralized matrix and the 4 different kinds of cells. The osteoblasts, which are in charge of bone formation, the osteoclasts, which resorb bone. Then we have the osteocytes, which are embedded in the matrix and communicate between osteoblast and osteoclast. And finally, we have bone lining cells, which are supposed to protect the bone surface while remodeling shouldn't occur. Bone, in fact, is always under constant remodeling, since it needs to adapt to mechanical loading, or for example, need to heal after fracture or micro cracks, and of course, to maintain the calcium and phosphate homeostasis. When we have cases of abnormal bone remodeling, these lead to several skeletal disorders, such as osteopenia, osteoporosis, and many others. Therefore, we consider any osteotoxicant that can be pollutants, molecules, and so on, which now I don't have the time to go deeply in each molecular pathway, but we need to just to keep in mind for today that osteotoxicant will lead to several problems, such as modifying the structure of the extracellular matrix, or impact the cell proliferation, differentiation, doing it directly on the osteoblast or osteoclast, for example, or of course also indirectly from other organs. As I said before, today I will show you some example of benzolfapyrine osteotoxicity, which I will call shortly BAP. BAP is a well-known compound from the polycyclic aromatic hydrocarbons family, and it comes from multiple sources. This can be natural, for example, it's found in the environment in the crude oil, but of course, many of them are from anthropogenic activity. In fact, BAP is normally formed during incomplete combustion of organic compounds, material. That's why it's ubiquitous and persistent. It is found in the atmosphere, in the sediment, and also in the water. And is also known to accumulate. Many people have already shown toxicity of BAP. I've shown that BAP is a teratogenic compound, carcinogenic, and also an endocrine disruptor. But still, little is known on its osteotoxic effect. So the objective of my past 2 years was to assess BAP osteotoxicity in zebrafish, trying to find insights into the cellular and molecular mechanism of this toxic effect on bone. In our lab, of course, we use zebrafish, a small fish coming from the east, due to all its technical advantages over many other animal models. And that's why today it's a good model for many kind of research, such as developmental biology, toxicology, genetics, and of course also for osteotoxicity. So these are the systems that we currently use every day in our lab. We can assess the effect of compounds during development, for example, looking at the operculum growth, or also at the development of the axial skeleton, and also the regeneration of the caudal fin and the scales. Finally, through some molecular analysis, we try to better understand the phenotypes behind this toxic effect. I will start now with Operculum, which was a system we developed a few years ago in our lab. The operculum is a flattened surface bone which covered the fish gills. It is formed early during development. In fact, we're already available to see it around 3 days post-fertilization, and of course can be easily monitored through imaging. Normally, what we do is to expose our larvae from 3 days post-fertilization, and at 6 days, we then stain them with alizarin red. And then through fluorescent microscopy and morphometric analysis, we measure the area of the operculum, and we normalize it by the area of the head to reduce interspecimen variability. So as you can see from this plot, what we did was to test many concentrations of BAP from 0.01 to 31.6 micromolar. And also we always integrate in our assay our positive control, calcitriol, or vitamin D, which is a non-osteogenic compound. So as you can appreciate from the images, for example, when we compare the area of the operculum in fish exposed to BAP, we can clearly see that the area was reduced. And after quantification of many fish, we could indeed confirm that BAP was reducing the area of the operculum at increasing concentration of BAP, suggesting that this osteotoxicant may interfere with the mineralization process. Then, to understand better how the cells were reacting to BAP, we selected 1 micromolar, which was the highest non-lethal concentration. And we have used two transgenic lines, SP7, which label in red immature osteoblast, and the osteocalcin, which labels in green mature osteoblast. And as you can see, we still confirm that the area of the operculum was reduced, the mineralized area, when compared to control. But interestingly, we saw that early osteoblasts were slightly increased upon BAP exposure, while mature osteoblasts were reduced about 60%, suggesting that BAP may affect also the differentiation processes during osteoblast maturation. Another system that we use, as I said, is the axial skeleton. Zebrafish, in fact, at 30 days present already all the axial structure mineralized, and we can stain them, the fish, for example, with Alcian blue and alizarin red, to then see cartilage and bone in the same fish. What we do in this case is to expose fish from 3 days post-fertilization, and at 30 days we double stain them, and through microscopic examination we look at the deformity number, typology, and also the area of the skeleton which are most affected. Here I'm showing you some example of the structure that we count as deformed. On the top we have fish exposed to control, and on the bottom to BAP at 1 micromolar. And normally we count as deformed structure, for example, partial or complete fusion of vertebrae, malformed vertebrae, fusion of emal arches, and also cases of platyspondyly, which is compression of vertebrae, or ectopic mineralization. After quantifying 100 of fish, we could indeed confirm with BAP that increasing the concentration was increasing the percentage of deformed individuals, reaching 100% at 1 micromolar. The severity of the deformity per fish also increased at increasing concentration, reaching the maximum at 1, where each individual presented more than 4 malformed structures in the axial skeleton. Looking at the area of the skeleton that were most affected, these were the caudal vertebrae, caudal fin region, and also the at the highest concentration of the caudal fin complex. Then we use also regeneration system. Zebrafish, in fact, can regenerate many organs, and the caudal fin is one of them. Caudal fin is also composed by segmented bony rays, and normally takes around 15 days to complete the regeneration process. This system is very interesting since it can give us information about the tissue regeneration upon exposure and also de novo bone formation. So what we do in this case is to use adult fish around 3 months. We amputate their caudal fin at day 0. We expose them for 5 days. And then after a laser staining, we look at the regenerated area, the mineralized area, and also we can do more deep analysis such as micro-CT to see the intensity of the rays, and also some morphometrics. Although in the case of BAP, we didn't see any effect on the regeneration process of the tissue, and also in the mineralization, we went through micro-CT to look deeply into each ray. And here I'm showing you the intensity of the ray from the amputation plane until a certain number of pixels. And as you can see, fish regenerating under BAP exposure were indeed presenting a reduced intensity of the rays. And after a quantification, we could indeed confirm that in the case of the caudal fin, BAP is also affecting the mineral density of the rays. Finally, through our last system, the scales, zebrafish can also regenerate scales. Scales are a bone-like mineralized structure that cover the fish skin. And as I said, they can also regenerate. They're very interesting since in this small unit, we already have bone forming and resorbing cells that we can assess. What we do is to extract scales from adult fish. We let them regenerate upon exposure. And at 5 days, we recollect them. And then through different staining that I will show you soon, we measure the scale area, circularity, the trap activity, and also the demineralized area. This is one example. On the left, we have scales stained with Van Cossa, which is a staining that stain in brown the calcium and phosphate crystal deposition. And on the right, we have a trap staining, which is an enzymatic assay, which will label in red the activity of the osteoclast. And as you can see, when we compare control scales versus scales that were under BAP exposure, we clearly notice already a reduction in their area, an affected morphology, and also an increase in demineralized areas and an increase in the osteoclast activity. After a quantification of many scales, we could indeed confirm at the different concentration that BAP was reducing the scale area from 0.1 to 10. It was also affecting the circularity of the scales at all concentration tested. The activity of the osteoclast was indeed higher when exposed to BAP, and in accordance, the areas of demineralized bone were also increased, suggesting that BAP also during scale regeneration may affect the remodeling processes. Then finally, we performed some molecular analysis to better understand what was behind this phenotype we just showed. And we performed some transcriptomic analysis of larvae that were exposed either to DMSO or BAP at 1 micromolar. And here I'm showing some pathway enrichment of the differentially expressed genes. Of course, as we were expecting, many pathways were leading to the xenobiotic signaling and metabolism, such as phase 1 functionalization of compounds, phase 2 conjugation of compounds, and so on. But interestingly, we saw that many pathways enriched were leading to extracellular matrix organization and degradation, such as we can see here, degradation of the extracellular matrix, collagen formation, and others. What we did after was to confirm by qPCR some marker genes member of these pathways. And to confirm the xenobiotic signaling pathway, we choose the HR and PXR, which are 2 nuclear receptors known to bind to xenobiotic molecules. Then the CYP1A, which is the cytochrome 450, which represent the first phase of detoxification of xenobiotic, and then also the glutathione S transferase, which represent the second phase. And as you can see, they were all indeed up-regulated and confirming the activation of the pathway. To then check for extracellular matrix organization and degradation, we selected MMP9s, which are known to be upregulated when we have a degradation of the extracellular matrix. And as you can see, both genes were indeed upregulated. And also other genes involved in the matrix formation and organization, which are collagen or osteopontin, for example, which were all down-regulated. And I would like also to show you that also OC2, which we saw at the beginning was reduced in the transgenic line, was also confirmed by qPCR. Therefore, what I show you until now is that BAP is really a strong osteotoxicant that affects bone formation, mineralization, regeneration, and remodeling. And we believe that BAP and in general other polycyclic aromatic hydrocarbon may affect bone formation through the HR signaling pathway or the xenobiotic pathway, which will lead to an inhibition of the osteoblast maturation or activity and a stimulation of the osteoclast activity. Okay, I will go now in the second part of the seminar, which is focused on the image analysis tools. Okay, as I just showed you, we perform our experiments, the experiments are done, but then how do we process our data? Especially when every week we end up with thousands of images of larvae, scales, caudal fin, and so on. So to analyze them, we use Fiji or ImageJ. These are two, it's a very nice software which runs everywhere in Linux, Mac, or Windows. It is open source, of course, and freely available to everyone. Behind has a big user community, which can help you if you have any problem. It works with many file formats, and of course has many tools such as image processing, analysis, editing, and so on. But what is interesting for me was that it allows us to create macros, which can automatize the image analysis. What is a macro? A macro is just a simple instruction that expands automatically into a set of other instructions to perform a particular task. It is basically code which will run during our analysis. But why macros? Well, as you may understand, is first to automate repetitive tasks. They can also be used to document what you do. And of course, also to share common procedures with your colleagues or other lab members. But most importantly, they have many advantages since they save our time that we spend at the computer doing manual analysis. They can standardize procedures and protocol. And they also reduce operator biases. They allow us to check also our analysis afterwards to see if indeed the measurements were well done. And they can also be adapted and personalized. 2 years ago, I was really struggling analyzing many images of caudal fin and operculum, and I was lucky to meet Fabrice, which is a bioimage analyst at the Bordeaux Imaging Center during an imaging course. We start talking about macros, and I could then apply to cost action to go and visit him in Bordeaux, and together we developed ZFBone, which, as you may understand, it's a tool set which was done to assess all the system I just showed you. And ZFBone, it's free available on GitHub, and we also made some YouTube tutorials for the people that are the first to see how each tool works. In ZFBone, we have different macros. We can analyze our operculum, for example, in the simple method, just the area of the operculum over the area of the head, or we can get the signal of transgenic lines, such as mature or immature osteoblast. We can use macros to assess scales parameters, to measure the caudal fin, and also to analyze the intensity of regenerated rays. Each macro has its own dedicated user interface, And here I'm just showing you for you to just have a look. For example, in the Operculum assay, we can choose if we want to analyze only one single image or a batch of images. If we want to apply some image adjustment, which channel do we need to select, and also which kind of measure we want. For scales also, we can choose if we want to measure osteoclast activity or demineralized area, and so on. And what is important that we also have, for example, in the threshold, we can choose if this will be interactive, that will give us the opportunity to stop and really confirm what the macro is measuring, or not interactive, and this will, of course, be more fast to get our results. Today, I don't have much time to show you in action each macro, so I will just show you the example of the caudal fin macro. And this is normally what we get from each fish at the end of each experiment. And normally for each experiment, we have like 50 individuals. We have a bright field image and a fluorescent one where the bone is stained with alizarin red. And that, as you understood, we need to measure the regenerated area, the mineralized area. We also take some ray morphometrics, such as the number of regenerated segments, the width, and the length of each one. And as you can imagine, this really will take a lot of time if we measure manually each segment and so on. So now I will show you shortly how the macro works, for example, during the mineralization assay. As I said before, here we want to first measure the length of the amputation plan, which will be used to normalize the regenerated area. And then we take the width segments before amputation to then normalize it with the mineralogenic area. And this is what we get at the end as output for each individual. So we are able to then to really check if all the measurements were taken. And we also take, we'll get a result table, of course, with our raw measurements and also already all the normalization done. So as you can see, you just need to select what to measure and follow the macro. The macro will stop at each step, allowing the user to select the region of interest. As you can see, some steps are still semi-automatic. They still require an user input, since between different labs and so on, it will be difficult to really automatize everything. And that's it. This really saves a lot of time, and also the data processing, it's automatically and done. Another example is to get the morphometrics of each ray. Imagine doing this manually, one by one, with the 16 rays and having 50 fish. So we created this macro, which require only for the user to set different landmarks between each segment. And automatically, the macro for each ray will report a table with all the measurements. As we can see here, each segment will report the width of each one, the length. And I also created a summary table, which will just report the average of interesting distances for each ray. This is the example of the output of the scale assay macro. On the left, we have trap enzymatic assay, and then on the right, Van Costa stain scales. And as you can see, what the user needs to do is just to select each scale, and then automatically the macro will get shape parameters, such as the area, circularity. We label each scale in the table, and also, of course, the amount of staining in each scale. We can do this also for measuring demineralized areas, such as in this case. And here in purpose, I just selected one scale, just to show you that this macro works with no matter how many scales we have, for example, in the images. Since we are here, I would like to show you also other example of macros that I developed during my PhD. For example, we use macros to detect and count cells. Here in this case, we are counting the number of neutrophils, and we just need to select a region of interest, and automatically we will get a result table with the number of neutrophil per fish, and also a screenshot to confirm if indeed all the cells were counted or not. And we can do the same for histological sections, such as counting number of goblet cells. In this case, we only need to select our region of interest, each villi, and then automatically the macro will give us the area of the villi and also the number of goblet cells in each one. Another example is, for example, to count and assess egg morphometric and their status. Last year, I had to quantify from 24 different corals the number of eggs, report their area, count how many they were alive, how many they were dead. So I did this macro in which, you know, if you put the threshold not interactive, you really don't need to do anything. You just select your folder and the macro will work for you. For each image, will give us a screenshot where the alive eggs are marked in green, dead eggs are marked in red, and also, of course, a result table in which we have each egg, their area, the circularity, and also how many eggs they were alive, how many dead, and the total amount. Another example of macro can be applied for fish tracking. Last year, we acquired in our lab this machine, the Zantic. This will track the fish behavior a long time. But we just got in trouble when we saw that we were just getting a set of coordinates. And we wanted to get more interesting data from this tracking. So we did this macro that first will show us the movement of the fish in each well. And from each fish, based on their coordinates, we can extract the total distance swimmed, the percentage of swimming time, resting time, average speed, and other parameters. Finally, this year, in collaboration with Elvira, a postdoc from the Aqua Group lab here in the CCMAR, we developed this macro which monitored the color progression over time. In fact, they were injecting some flat fish with the different colors, and we wanted to see the color how would fade over time. So what I did was to develop this macro, which first we select our region for each fish, then the macro will align each mark. For example, here we had 15 fish, so this will be aligned in a stack. Through a threshold, the marks will be recognized automatically, and then we can get, for example, the main saturation, which was used to then estimate how the color saturation decrease along time. So that's it for today. Thank you very much. And my take-home message is save your time and just run a macro. And I would like to acknowledge, of course, the CCMAR for giving me the possibility to present my work, all my supervisors and collaborators, and of course also the founding agency that are helping me during my PhD. Thank you very much. And I'm ready for questions.
Thank you, Mark, for your kind presentation. Now it's time for questions.
So I have one question. Imagine you don't know anything about computing. How hard is to know to learn how to do a macro?
Well, first of all, of course, it's not easy. You need to understand a little bit of coding. But for example, in a Fiji website, there is already some guides to give you a starting point to start and also writing your macros. Fiji has also the possibility to record what you do during your analysis. That of course needs to be adapted, but still can be a start for creating small macros, for example.
Okay, so you can go to Fiji and see the links for that, and you can record. After using the record function, you can also edit those macros, no? When you start.
Yeah, so already from Fiji, before starting an analysis or a workflow you want to do, you start the recorder. He will record all your steps. And then from that is already a macro that of course will need to be adapted then to work with the different images and so on. But already for some simple steps, it's already useful. Then on Fiji website, you have a page where you have all the function described and what they do. So then of course you need to spend a lot of time, a lot of nights writing, testing.
Okay. Thank you very much. It's very nice.
Thank
talk.
you.
Marco, do you hear me?
Yes.
Are all these macros available on the internet or only the ZF bone macro?
For the moment, they are available only the ZF bone. The one in collaboration with Elvira, the paper is under review, so hopefully soon it will be available a tutorial online on YouTube and also a GitHub page. And now I'm working on publishing the other macro, the one for the behavior, eggs, and goblet cells.
Okay.
Any more questions? I have a question, Mark. How easy is to adapt one of those macros that you show, for example, to count, for example, cells, other kind of cells, or moving cells?
Sometimes it's very easy. We just need to maybe add or cut some steps, but then the main processes are normally similar. So then, of course, the same macro I used to count neutrophils, I use it also to count, in another case, microplastic particles or other things. So with sometimes they can be adapted and be applied for many, many things.
Thank you. Any more questions?
Hello, Marco. Maybe a tiny one. Tiny little question. So it has been a huge amount of work on the macro and really great presentation. But
Thank you.
What will come next? What macro are you thinking about? Well, what is the next step?
Well, the next step, in my opinion, for example, at ZF Bone, we have already starting to work to transform the Operculum macro in Java, which took a little bit more time than do a simple macro. But of course, then in Java, we're able to do many things and can be even become independent from Fiji, for example. So my dream will be to create my small software for assessing, for example, bone.
Excellent, thanks.
So I think there's no more questions. So before we finish, I would like to remind you all that now all the CCMAR seminars are disseminated through the YouTube platform, and If you are not able to make your own registration, you can use our YouTube channel, the CCMAR YouTube channel. And I also like to encourage you all to subscribe to our YouTube channel. Thank you all. And thank you, Mark.
Thank you. Bye-bye.
Bye. Thank you very much, Marco.
Thank you, Claudia.